Okay, go ahead. We are recording.
Okay, great. So I want to welcome everyone. This is to the kickoff for this year's Biomedical Cross-disciplinary series. And the goal of this seminar series is to promote networking and collaboration in translational health among researchers, healthcare providers, and policymakers from different disciplines to shift the paradigm from seeking a cure to developing a strategy of prevention. This year's topic is metabolic syndrome, and the series runs through May. Today, Dr. Loretta DiPietro will be presenting on the public health aspects of metabolic syndrome, specifically aging, insulin resistance, and the role of physical activity in risk mitigation. Dr. DiPietro is an accomplished and widely published researcher with particular interest in the role of physical activity in the health of older adults. She's been awarded grants from the NIH, specifically the National Institute of Aging, as well as the American Cancer Society, and has lectured at medical schools, public health schools, and other organizations around the world. Dr. DiPietro is a full professor at the George Washington School of Public Health. Prior to that, she was associate professor of epidemiology and public health at Yale University School of Medicine and a fellow at the John B. Pierce Laboratory, which studies how biological systems interact with the built environment and their influence on health, which we talked a little bit about earlier. If you want a little background, the School of Public Health is fabulous in part because of Dr. DiPietro. Anyway, recognizing that many of today's critical public health problems can be addressed at least partly through improved nutrition and physical activity, Dr. DiPietro has built bridges between population-based public health and the clinical and psychological domains of exercise science. And we look forward to hearing about those connections from her now. Welcome, Dr. DiPietro.
Well, thank you. Thank you for that introduction, and thank you for inviting me here to do this. I'm just getting set up here. Okay, unless I hear otherwise, I assume everyone can see this. All right, so as mentioned previously, I'm speaking about insulin resistance, metabolic regulation, glucose regulation, but particularly how it affects aging people. I mean, we're all aging. I shouldn't have said that. I mean, how it affects old people, right? And oops, let's turn off the phone. So let me get started. I've spent a career studying physical activity and its health effects, and most specifically in older populations who really are quite vulnerable. It shouldn't surprise anyone here. Here I have the prevalence of diabetes in the United States by different age groups. And you see this dose response relationship over here in the prevalence, with increasing prevalence as people get older, right? And there are a number of reasons for that. And we'll be discussing those reasons, as well as ways of protecting older people from this metabolic dysregulation that we observe. So a lot of this decay or derangement that occurs in older time has to do with physical activity and movement, right? So the modern day lifestyle, as you all know, is characterized by a majority of time spent sitting and reclining throughout the day. And this is particularly true for older people, right? And in fact, we monitor older people all the time. We know what they're up to. Older people may spend up to 60 or 70% of their waking hours sitting or reclining. And it's not just older people, people over age 60 or 65 that do this. Most Americans may not admit it, but spend most of the day sitting. However, older people might be especially vulnerable to the deleterious effects of prolonged sitting, because we all lose physiologic reserve as we get older. So any perturbation has a bigger consequence in older age. So there's particularly susceptible. So a talk about insulin resistance really goes back to 100, 1,000 million years ago, right? And how we were designed. We are designed to be hunters and gatherers, and move in order to obtain food that sustained us to then get up and do it all over again. So, and traditional hunter-gatherer lifestyle was characterized by what we call a subsistence efficiency ratio, meaning the ratio of the calories that they needed to eat in order to sustain the work output needed to gather the food they needed to eat to do the work. Eating and physical activity are linked, right? And so if we go back to hunter-gatherer days, based on records, from teeth and bones and stuff, the subsistence efficiency ratio back then was approximately 3-to-1, meaning people took in 3,000 calories and to support the work output of 1,000 calories per day. Okay. Now enter the modern age where hunting and gathering means going to the refrigerator, basically getting out of the recliner and going to the refrigerator. You can have your food delivered to you. I mean, we don't have to work in order to eat any longer. Consequently, and especially in the last 150 years or so, energy intake has gone down a little bit overall. However, we're putting out about 300 kilocalories of work or activity output, right? So this is on average, and this is a subsistence efficiency ratio of 7-to-1. This is why we have diabetes. We are designed for an efficiency ratio of 3-to-1. Modern day has put it approximately at 7-to-1. Well, what does that mean? That results in an overfeeding of the cell, right? Excess nutrients come in and they're not used for fuel. They build up in the cells. And so if you look at this cartoon, right, and we all have metabolic challenges occurring every single day, right? But this overfeeding, right, especially overfeeding with fats, et cetera, along with aging, causes this, we call it substrate buildup in the cell. And consequently, your free radicals build up, affecting mitochondrial function and insulin action in the cell, right? So this substrate buildup will decrease adipogenesis. It increases lipolysis, right? And it decreases fat reesterification. And the consequence of all of that is insulin resistance and type 2 diabetes. So this is how I simplistically categorize diabetes. It's an overfeeding of the cell that interferes with insulin action and mitochondrial function in the cell itself. So when we study insulin and insulin action, there are a number of places and ways that affect insulin action and glucose metabolism. With physical activity, my area of interest, I'm not really a physiologist per se. I do some physiology, but I study it, number one, at the level of the insulin receptor, right, which exercise is tremendously effective at cleaning out the gunk from the insulin receptor. If you think of a rusty lock that you can't get your key in, right, exercise is wonderfully efficient at getting rid of that crud so that the insulin molecule can bind to its receptor, right? And the other area of interest for me is this glucose transport protein. What muscle contractions do, and we'll talk about that later, is the transporter proteins will come to the surface of the cell and escort the glucose right in. Right. So there's two mechanisms. There's the insulin receptor, but then independent of that, there are muscle contractions that will dispose of glucose without insulin, and that or independent of insulin. So that's where physical activity does its best work. And older people who exercise on a regular basis, they're protected from the metabolic dysregulation that leads to insulin resistance, type 2 diabetes. Remember, insulin resistance appears before diabetes. And so you can correct that fairly well with exercise. And what we're trying to do is prevent people from making that leap to type 2 diabetes. So older people who exercise are more protected than those who don't. But compared to young people, the responsiveness to exercise training is a bit blunted in them. And again, they have a lower physiologic reserve, a lower cardiorespiratory fitness, lower strength. So many times the stimulus given, is much smaller than the same stimulus given to a younger person. Nonetheless, we can improve their insulin sensitivity quite effectively. And the magnitude of improvement, as I said, number one, has to do with their biological age or their fitness status. Also has to do with initial level of impairment. We all know we see the biggest, strongest improvements in people, especially older people, who have the highest initial levels of impairment. I mentioned just a few seconds ago the differences between absolute intensity and volume of the training bout. And then also the thing that people forget about is, the timing between the last bout of exercise and the assessment of, glucose level, insulin levels, free fatties. Right. So the timing of when you take the measurement is also very important. You're going to see that in a second. So my goal today simply is to describe some earlier research of mine, because you asked about insulin resistance. And so number one, to describe enduring effects. Everybody knows you can improve metabolism with every exercise session, right? You get this transient improvement. But at the time, I was interested in, how long do those effects last, and can we prolong those effects? So I want to describe the enduring effects of exercise intensity on multi-tissue insulin resistance. And I also want to show you some short-term effects of exercise of different types and volumes on glucose tolerance itself. And then in some later studies, I'll describe the effects of timing and dosing of exercise, because, like a pharmaceutical study, we now have sufficient information to affect the timing, just like the timing of medication use. So we'll start with the multi-tissue insulin resistance, 72 hours post-exercise. Metabolic control in older age is a function of, three primary areas. The first that I'm talking about is this multi-tissue insulin resistance. I haven't mentioned here the drop in sex hormone levels leads to this preferential deposition of body fat. You know, and it will accumulate not just centrally in the abdomen, but it will accumulate in the visceral depot, which, if my cursor would come around somewhere, is this is the deep abdominal depot, comprising hypertrophied fat cells that are quite metabolically active and you know, do some damage metabolically. This is the visceral adiposity. The good news is this is the first fat to go with exercise. It's the first to metabolize, right? And so we'll talk about this because of that, and we'll also mention beta cell function in older age. So these are 3 sites we studied of insulin resistance. I just told you about the visceral adipose depot up there in the top right. Well, those fat cells drain right, you know, right through the portal vein into the liver. And so excess mobilization of fats and a fat buildup in the liver will interfere with insulin action. So that's one site of insulin resistance. When fat cells, you know, reach their limit of size, the fat will spill over in other areas, most notably muscle tissue. Right, and that is the major site of insulin resistance in people. And then the fat cells themselves, peripheral adipose tissue, will become resistant to insulin with regard to reesterification. So these are all women. I study, they're really older women, if you look at them, with big middle areas. Their average age in this study was 75, and no hormone replacement therapy. And that's, you'll see what happens later with that. We exercise them a number of different ways. I'm just showing you the overall study protocol. Right, baseline measures. This was nasty. This was about as invasive a study as you can get, right? So lots of screening and measurements of fitness, glycemic control, CT scans, DEXA scans, the euglycemic clamp for the gold standard of measuring in peripheral insulin sensitivity, muscle biopsy, bloods, blah, 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 right? They train for 9 months, 4 days a week for 9 months, 2 aerobic training groups and 1 resistance training group. Aerobic worked at either at 80% or 65%, so a moderate intensity and a higher intensity training group. Pay attention here to the follow-up, because remember, I'm talking about 72-hour effects. We repeated the euglycemic clamps like on the 4th day after their last exercise bout. And so this is what it took with regard to duration. These women were in, I held caloric expenditure constant at 300 calories per bout in the 2 aerobic groups. And so obviously it took the group doing moderate intensity activity a little bit longer to complete that. But this is a good old bout of exercise for 9 months. This is the clamp for the protocol for the clamp. As you see, blood draws every 5 minutes, little blood draws. infusion of different isotopes to measure hepatic insulin sensitivity, and to measure turnover, glycerol turnover in the system. Two levels of insulin infusion, a low dose, right, which tells you a lot of things, and then a high dose, 40 milliunits, which is perfectly physiologic. It's what, you know, after you eat Thanksgiving meal and you're about to explode, that's about where your blood, this infusion mimics a big meal. So it's nothing, you know, extraordinary. All right, so the first set of data that I'll show you are some characteristics before and after training, right? So this is the age group. The first thing you're going to notice is that their level of aerobic cardiorespiratory fitness did not change one bit after 9 months of training, right? And every time I show this slide, all the exercise physiologists think, oh, you're not measuring it correctly. We know how to measure it. These are women not on estrogen replacement. And if you separate out people's data and only look at women, older women not on estrogen replacement, you'll see how difficult it is to increase aerobic capacity in older age, right? Most of the improvement in aerobic capacity has to do with peripheral improvements, which don't in women, versus men who seem to improve more centrally. And this particular test doesn't pick it up, right? And also keep in mind, even though there were, some of them were exercising at 80% of their maximal aerobic capability, you know, their aerobic capacity is low in older age. So 80% of low, like 20, look at 21 milliliters per kilogram of body weight per minute. You know, this, even this here translates into an absolute intensity of like 4.5 METs. And so at that age, that's probably not enough to, you know, really significantly improve aerobic capacity. The problem was we used to think it was necessary to improve metabolic regulation. Here's body composition in this group. Once again, you would never know that a 9-month intervention occurred, because look at just BMI, right? Very little movement. Lean mass, very little movement. Some, you know, increases, but nothing to write home about, you know, 9 months, right? Body fat didn't change at all, whole body fat. The needle moved a little bit with regard to the abdominal circumference, meaning any fat loss was coming from the visceral depot, more than likely. So imagine after, you know, 2 years, these results come in, and I thought my career was over until, you know, the clamp data got analyzed. So from the euglycemic clamp, as I said, you calculate based on infusion of, you're loading in insulin. Well, you better also be putting in glucose or the person dies, right? And so you measure insulin sensitivity by the glucose utilization rate over that 2-hour, 4-hour period. The pink bars are the low dose insulin, but what for insulin sensitivity, you really want to look at these lime green bars. This is percent change. And the first thing you see is this beautiful dose response curve between like intensity of training and improvement. Keep in mind, this is 72 hours after the last bout. So everyone improved. Here's a 7% improvement. Clinically, that's quite meaningful. This is a 16% improvement, and here was the 21% improvement, which achieves statistical significance here. With regard to insulin sensitivity in the liver, we measure that by hepatic glucose production. Again, if we're infusing insulin, that insulin should halt hepatic glucose production, right? It should suppress it. So you're measuring the insulin sensitivity there, and it's a little hard to see, maybe not, but the gold are significant findings. And so at least in the moderate intensity group, we were able to significantly improve hepatic insulin sensitivity. That might be because they started out much more impaired than the other group. But in any case, we normalized it, both at baseline and at low dose insulin. So that was good news as well. And then with regard to glycerol turnover, we observed a significant reduction at the low dose insulin with higher intensity training. But again, it might be because they started out higher. Their level of impairment was higher. The low dose insulin is really what you want to look at for, suppression of free fatty acids, hepatic glucose production, things like that. For muscle insulin sensitivity, the 40 milliunit clamp is better. So just to summarize from those experiments, there was no change in VO2 or body composition. These are the two hallmarks that people would use to say, well, a training effect occurred and then health improved. These were independent of those two things, right? And glucose utilization improved and was evident 72 hours after exercise, which is impressive. And there was also improvements in hepatic glucose production and glycerol turnover. With higher intensity activity specifically. Well, this has implications, right, for how we might prescribe exercise. So if you can get these enduring effects, that might mean maybe you don't need to work out every day. Maybe you can get by with 1 or 2 days, but in order to do that, you've got to exercise at the load that this group did. All right, so now let's talk about some transient improvements in glucose tolerance, right? That involves insulin sensitivity. I put two groups together and I compared the low volume, lighter resistance activity against high volume, moderate to vigorous intensity activity. So same study protocol, but now I looked at findings from the oral glucose tolerance test, and we did this within 2 days of their last bout. In fact, mostly the next day. So here are the glucose curves. If you're familiar with a glucose tolerance test, you have the, you take baseline samples, and then they drink this sugary stuff, and it's a known volume and concentration, and then you sample blood over the course of 3 hours. And so you get these lovely glucose curves and insulin curves. We also measured free fatty acids. I saw the opposite of what we observed at 72 hours. How could this be? These are the lower intensity resistance training on the left. And if you look at these curves, here's another beautiful dose response relationship between months of training and flattening of that curve. The baseline curve is on the top right here, and especially, the back part of the curve comes down, 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 meaning they're coming back to baseline much faster in a graded progressive manner. That was beautiful. And you see the same thing with the insulin curve down here, the lower left. Here's baseline, the dotted line. I lost the cursor again, and it gets increasingly better, indicating improved insulin sensitivity. Well, look on the right side, the exact opposite happened in the high volume, moderate to vigorous intensity group. First of all, their glucose looks completely impaired, number one, and then very little change, no change at all over 9 months. Their insulin got worse. This is baseline down here, that the dotted, and over the course of training, they actually became, or at least when their blood was measured the day after exercise, they look impaired. I thought, how could this be? Right? If you like looking at bar graphs, here is the late phase of the glycemia curve, the last hour. And you see this in blood glucose levels progressively decreasing. And at the same time, insulin in the last hour is decreasing over time. So you have better glycemic control for a lower insulin cost. That is real improvement, right? Whereas at 24 hours, look at this group, no real changes in glucose, and the last part of their, the last hour here, you see increasing levels over 9 months. Hold on 1 second. So again, I'm thinking, how could this be? And so I went to the marathon literature, and sure enough, found a paper, of marathon runners who are tested, undergo metabolic testing the day after a marathon, and they're all impaired, right? High levels of fat in the blood, insulin resistance, high levels of sugar, you name it. And I thought to myself, well, maybe this large volume of moderate to higher intensity exercise is like a marathon for a 75-year-old person, right? And we all know, with excessive, or unaccustomed exercise, or heavy loads, there are a number of stress and counter-regulatory responses that occur in the short time, right? So, cortisol increases, stress hormones increase, growth hormones, inflammatory factors increase, levels of free fatty acids. There's a, evolution did this to us. It ensured enough fuel to remain in the system under stress to outrun a saber-toothed tiger. Well, what if this load were like a marathon? And so I just draw this hypothetical thing here. The solid pink lines are young people, right? So maybe, these marathon runners, they show initial impairment, but then later on, the impairment resolves itself, and then there is a period where you would observe the improved insulin sensitivity. Maybe this dotted line are what older people go through, right? The stress response maybe lasts a little bit longer, but yet is replaced by improvements like at 72 hours, where we saw, this is just my thing, my way of explaining it. And I told you we collected free fatty acids in the group. Insulin secretion from the pancreas should have an effect at lowering free fatty acid concentrations, right? And so insulin will suppress. Insulin is a storage hormone, right? So, and you see that here's baseline, the dotted line, and then training, after training. And in the lower intensity group, resistance training, you see in the first 30 minutes of the oral glucose tolerance test, which is when insulin should, in a healthy person, start to spike, you see this suppression of free fatty acids. But over here on the right, here's baseline, the dotted line, but after 9 months of training, at 30 minutes, there's very little suppression going on, right? It takes much longer for it to drop down. So these are stress effects of, these large bouts of moderate to vigorous physical activity. So, to sum here, we observed a greater short-term effectiveness of lower intensity resistance exercise, right, compared with the high volume moderate to vigorous. And this might be explained by the positive changes occurring in muscle quality from the resistance training, and these positive changes in quality, biochemical responses, greater perfusion, capillary density, were not hindered by this persistent stress response. And then the higher circulating levels of free fatty acids more than likely interfered with the insulin action in the liver and the muscle. And so what we started talking about was the metabolic benefit to stress ratio of the exercise among these older women. And again, this could affect how exercise is prescribed for older people. Now, I talked about these two areas. The third area that interferes with glycemic control in older people, it's not so much insulin sensitivity or insulin resistance, but it's impairments in insulin secretion, right? And due to aging, a loss of beta cell function with aging. So some older people, they may be very insulin sensitive, right? They're muscles because maybe they move around a lot and lift weights, and but they still may show signs of impairments in fasting glucose or postprandial glucose because their pancreas is slow, and they show a blunted secretion, delayed and blunted. And this is especially happens toward the end of the day. So I did one little experiment in 4 older runners. These are all masters runners, very healthy. They run marathons. The average age was like 68 in this group. And I brought them in for 2 separate oral glucose challenges. One, when everybody in the world does them at 8 o'clock in the morning. And then 2 weeks later, sorry, again. Okay. Then again, 2 weeks later, we repeated the challenge under the same fasting conditions at 6 p.m. So in these 4 people, over on the left is glucose. The solid line is a morning test, and you see how healthy they are, right? Like their glucose goes up, comes right back down in 2 hours. Beautiful response. Then when we tested them at 6 in the evening, look at the dotted line. Look at their glucose started at the same exact spot, but look what happens. It's like they're impaired over 2 hours. And the reason for that is, look over to the right here. Here's the insulin response in the morning. This is such a robust response, a testimony to how healthy these older people are. They're all men. And insulin was doing its job. But look at their insulin response in the evening. It's like non-existent. It's not only delayed, look at how blunted it is relative to the morning. So if this is happening in master's level marathoners, what's happening to us? I say I'm over 60, right? What's happening to our mothers? What's happening to our grandparents, right? And, the older, I told you about the prolonged sitting increases with older age. Well, what's the biggest meal of the day that people eat? It's dinner. Then you stumble over into a recliner to watch the news, Jeopardy, Wheel of Fortune, and Dancing with the Stars, right? That's the worst thing you could do, right? So how can exercise help these people? I don't see much evidence that that exercise can, fix the pancreas, right? But we realize that a number of other benefits of exercise that I mentioned. Just muscle contractions all by themselves will stimulate the GLUT4 transporter proteins to the surface of the cell to pull in the glucose molecule. Both insulin and muscle contractions will do that. Together, they're additive, but they can also do that independent of each other. And also, exercise, independent of insulin, enhances capillary perfusion, increases the surface area for glucose and insulin interaction with the muscle. So muscle contractions all by themselves can stimulate insulin action in the muscles independent of any endogenous secretion. So how about we add exercise, to an already sort of still working pancreas, but just to give it, to help it out, to supplement what's being secreted. So why don't we apply exercise, right? And to the period of time when older people are most vulnerable, and that's postprandially, right? And so how about we, the traditional dose of exercise that people are prescribed usually is like 45 minutes of walking. That is the most accessible prescription. So, okay, let's chop it up and apply it after meals, right? And so the muscle contractions can help clear the blood glucose out because the pancreas isn't working well enough. So we proposed that post-meal walking would be better at clearing glucose compared with one big large dose, right? So we split, this is another grant, a low intensity walking, right? But we timed it a half hour after each meal. That's, you know, the period of absorption. Very similar to a drug trial. Here, I was able to use the metabolic chambers up at the USDA. And so older people came into the chambers for 48 hours, right? And their first day, they just sat all day. And then on the second day, they either got post-meal exercise, a 45-minute bout of exercise walking in the morning or in the afternoon before the evening meal. So you got a number of different controls going on: sustained exercise versus post-meal, post-meal versus pre-meal. We monitored glucose continuously with the glucose monitoring system. They're very common now in research. Here's what a whole room calorimeter looks like up at the USDA, bed, treadmill, so we could set the exercise challenge. A sleeve for taking blood draws over the course of 2 days, and meals are delivered. And here are the findings from that. The first set of findings have to do with overall 24-hour glycemic control. And I'm comparing the exercise day to the control day in light gray. And what you see here is that both 45 minutes of walking in the morning and the post-meal exercise were somewhat equally effective at lowering glucose throughout a 24-hour period, right? The post-meal actually did a teensy bit better, but they were both effective, not the pre-meal late day exercise. But here were the most startling effects. And first, I want to direct you to the 3-hour post-meal. Remember, I said this is when people, especially older people, are most vulnerable after the evening meal, right? So the morning exercise obviously did nothing to help out 3-hour post-meal. This is 9 o'clock at night. And neither did the pre-meal exercise because glucose was higher. But the third set of bars, this post-meal exercise, post-dinner exercise significantly lowered blood glucose compared with the control day. This is where it counts, right? And it was very, very effective. If you look over here at lunch, post lunchtime, so this would be about 3 o'clock in the afternoon, morning was a little bit effective. Post-meal was effective. This was borderline statistically significant. But I call your attention here where on the second day, blood sugar at 3 o'clock in the afternoon was significantly higher than it was the day before. This is before they did that 45 minutes of walking, right? So this essentially represents 36 hours of sitting or reclining. And look at the damage that gets done. I'm only saying that because we all binge on Netflix. Here's the kinetic data, right? These are the 3 meals, the peaks that you see, and this is blood glucose. And I just drew a circle around the evening because the post-meal exercise here, this line, and not only here, but with each meal, significantly blunted that glucose excursion, right? As soon as they start exercising, down it comes, right? Disposed. And especially here after the evening meal. And the benefits lasted through the early morning. So if you can't do 3 bouts of post-meal exercise, we tell them you walk after the evening meal. Italians have been doing it like forever, since day 1. So to sum up this entire talk, the benefits of high intensity, high volume exercise are more enduring. They last longer. But lower intensity activity is less stressful, but the decay is more rapid. So if you're partial to lower intensity activity, you have to repeat it daily to maintain the benefits of it. And then short bouts of lower intensity activity that are perfectly timed after a meal are extremely effective for maintaining glycemic control. And this is so especially late in the day. Why is this important for public health? Because, most older adults do not meet US guidelines for physical activity. The majority of them, like 65, 70%. And given the growing prevalence of diabetes in older age, and what we know about the effectiveness of muscle contractions to prevent and delay the diabetic downfall. These findings have tremendous, I think, clinical and especially public health relevance to how exercise is prescribed for older people. So I'll end there. Thank you. Thank you to all my colleagues in and around. And I think there's some time left for questions.
Well, thank you. That was wonderful. And we have some time for questions. You can go ahead and put them in the chat, or if you'd like to, it's a small enough group, but I think you can go ahead and unmute and you can just ask the questions that way. I have a bit of a comment. I love this work so much, and I love that it's finally kind of catching on. At nutrition conferences, we're starting to see a lot of this, and we're calling it chrononutrition, right? It's part of this, that the timing of food matters, and the fact that you now have not only, okay, well, we could eat things at a different time, or we could eat them at the time we already eat them, and then we just go for a walk afterwards. Right. I love that fix.
Right. Like, I don't eat ice cream at night. Right. I have this like pancreas. I'm extremely insulin sensitive. But some genetic thing in my family, my pancreas doesn't, it's not as robust a response. So every time the physician draws my blood, it's like, oh, you have pre-diabetes. And it's like, no, I don't. Right. So I make sure before I go have my blood drawn, there is no wine at night, and I run over to get my blood drawn. And that just took care of that immediately.
That's great. Yeah, I have a similar habit. I've actually been discussing this with my husband a lot lately, and I tell him, you probably notice that I'll eat pancakes for breakfast, but I wouldn't be eating them for dinner. Right. It's having that amount of carbs for breakfast is, well, maybe not the best rule, but it's totally fine. Whereas with dinner, you would see very different results.
Right. Well, think of pasta. Right? A big pasta dinner. Probably not the best time to be loading up on pasta, and usually a bottle of wine goes with that. So, I mean, think of how we eat. But okay, do that, but you got to move around. And so what do we tell older people? You know, and they say, well, it's winter time. I'm not going to get, well, you can walk inside for 15 minutes. And while you're watching Jeopardy, every commercial, you can, you know, get up and march in place. That's not hard, you know. So there are ways of getting around it. And okay, you don't want to walk for 45 minutes. We now know, not just for glycemic control, but all kinds of risk factors, that you can chop it up over the course of the day. It's like a glass of water, right? Just over the, or whatever, just it's the volume over the course of 24 hours that matters. Hi, Brett, it looks like you have a question.
Yes, excellent talk. I really found this fascinating. I'm wondering if you can elaborate a little bit more on what the same energy output from the initial study, since that was controlled, what that looks like for people at different ages, different genders, because it was really important to control. I think it was 300 kilocalorie output for all of that, for the exercise. What does that look like for a 20-year-old male, 45-year-old female? You know, how much does that change?
Okay, right. So I can quickly pull up the slide again and show you. So can you see this down here? Right. So all groups trained 4 days a week. Oh, sorry about that. What did I just do? So for 300 calories. So let's say, you know, it depends on their body weight and their level of VO2 peak. So these are little old ladies, sort of, and they have a baseline VO2 of about 1.4 liters, right? This is a hypothetical thing. So for someone with a VO2 of 1.4 liters per minute who weighs 68 kilos, it'll take about 55 minutes at 85% VO2 or 65 minutes at 65 to expend 300 calories. So if you are a man who, with a younger man with a much higher VO2 max, and maybe you weigh 75 kilos, you can expend 300 calories in what, 15 minutes, maybe 15. So it depends the time. So think about the poor little old lady with a very low VO2 peak, right, of like 1.2 liters, who weighs 50 or 45 kilograms. I had one woman, honest to God, 80 minutes to do the moderate intensity to expend that many calories. She did it. She came. The adherence, retention was 99%. Adherence to the high intensity training was 95%, and it was 90% for the moderate, meaning they did all the sessions. And, you know, higher intensity, you get it over and done with quickly. That's why I do high intensity training. You're in, you're out, 30 minutes, bam, you're done.
Me too, Loretta.
Yeah, right. And the moral there is there's no better time to start than now to get some of these benefits, because it will probably only get harder to reach, you know, more input to get the same benefit.
Yep. That's why aging is a life, successful aging starts in utero. It's my new motto. I mean, the way pregnant women exercise now, there's evidence affects not just the newborn, but the programming of that newborn over the rest of their lives. It's a lifelong thing. And so, I mean, yeah. And we're hoping for the day where, clinicians make it, like point of care, asking about physical activity, right? They ask me how much I drank, do I smoke, take my blood pressure, put me on a scale, my height. Nobody says a word about physical activity. Right? I mean, that's not that
except
common.
for the pulmonologist. That's the only person who asks.
Right. Okay, so wouldn't it be great if everybody had a Fitbit and it synced with your electronic medical record?
Oh, that
Or
would be great.
at least ask about it. How many hours a day do you sit? Anyway, that's my shtick.
Well, thank you so much. I really appreciate it. You have such a wealth of knowledge on this topic, and I think you also make it digestible, right? These are things that people can do in small chunks. It doesn't have to be the 80 minutes straight workout, which doesn't sound doable at all. But if you break it up during the day, okay, I can do that.
Yeah, nobody does. One of the biggest criticisms of this study was, that's not feasible. Nobody's going to go exercise. So, the translation of science into practice doesn't always go smoothly, right? But I was very happy with the findings from the post-meal thing. That has the most applicability. But even then, people criticize. Nobody's going to walk 3 days a week, 3 times a day. Nobody's going to do that. All right. Okay, go take a pill. Go take a pill then. Yeah.
Right. Well, I think it's definitely a good goal to have, at least, right? Even if you aren't getting quite there, having this as a goal is a good place to start.
Yep. Just every step counts.
Exactly. Well, I think that's the perfect way to end. Thank you so much for joining us, everyone, and have a great rest of your evening.
Thank you.
Thank you.
Hey, Lee, do you have control of the meeting?
Let me look.
Because
it says I'm a co-host.
Yeah, do me a favor and end it. See if you can end it.